I am Dr. Jennifer Grier. I'm an associate professor at the School of Medicine Greenville, which is part of the University of South Carolina system of institutions. So here I am going to show you a little bit about the Echo Revolve Microscope. So this microscope is a really cool tool because it allows us to look at cells from a variety of different perspectives. What I'm showing you right now is these are some cells that grow in a dish. This demo is not alive, but we could very easily do this with some live cells. And what I'm able to do here is I can control what I'm seeing on the microscope through this iPad. Saves us a lot of effort from having to focus into small eyepieces. It's really easy for students to understand it, and it's a really great way to capture images that then I can take and analyze on my computer really easily. So here's an image, and if I wanted to just take that image, I can turn it on and off. If I need the light on, there we go. And then if I want to take a picture, I can just press. Now I have a nice photo. But one of the cool things about this microscope is more than just doing dishes, I can also look at microscope slides. So one of the major benefits is if I take this off, I can remove the stage here, set that off to the side, and then I can come around and there's a button on the back. If I press this button, the entire microscope turret will flip to right side up. Now I'm just carefully going to add the stage back on. Oops. There we go. Flip it on. And now I can look at cells on a microscope slide. And this will allow me to get very, very close to the slide, which means I can see my cells at really high magnification. So let's see what we can find. All right. So here we go. We can see some cells on the microscope. And because I've done this on a microscope slide, I can actually get a lot closer. Let me turn that exposure down so they actually look pretty good. One benefit of this microscope is it's actually telling me where you see white, things that are overexposed. That's important because if I wanted to analyze this image, I need everything within sort of the detectable levels. The ones that are showing white, it's way too much. So I can fix that by just reducing my exposure. And if I go too far, I can bring it back up a little bit. Now I've got a great picture. Each of these blue dots is the nuclei of a cell. And so if I like this photo, I can go ahead and capture it. And then I can move on and see some of the other colors or things I might want to detect. So in this case, green is a protein of the immune response. So you can see this one cell up in the corner has a really strong response. The one in the middle maybe slightly less. So let me capture that because I like it's neat to see that. I can also, here we've got another color in red. This is a measure of stress. So we'll capture that. And then lastly, some virus proteins. I'll capture that. And so what we have here is now an overlay where all of these different colors that I've collected are visible. And I can come off to the right here and turn on and off some of these different images. They're all just overlapped to see the different components that might be interesting. So I can see which cells are really bright and red. I could turn off red, turn on green, see where I'm seeing green. So this gives me ability to look at up to 4 different colors of fluorescence, meaning I can look at 4 different proteins or markers, get a lot of information from these cells. We're only at 40x, we could go up to 60 or 100x, so we can get even closer. I could even, this is nice, I could zoom into those cells on the screen but I could also take a closer magnification on the microscope. So we've got a lot of flexibility here. And the other benefit is this is really user-friendly. Students are able to come pick this up, understand how to use it really quickly, and because it's run entirely through an iPad, anything that an iPad can connect with, this can connect with. So we can use this to connect with the big screens that we have in our classroom and send these images up to these large wall screens so every student in the class can see them. They don't have to be directly in front of the iPad. So this is a great tool that we're working on using at the School of Medicine Greenville. We had an opportunity to take a lot of photos this summer, and I can just show you some of the ones that we had a chance to take. So this was a fungus that we found growing in the lab. Not a great experience to find fungus when you didn't want it, but really great photos. And this helped us identify what was going wrong. So that was really nice. We also have some researchers that were working with sea urchins. And so they were able to, what you're seeing in green is calcium within the sea urchin. So they're able to model and see how calcium is moving during sea urchin development. And in fact, some of these are 2 and 3 cell embryos, which means they're already starting to divide and grow. And with studying sea urchin embryos, we also look at sea urchin sperm. So this is an example of sperm from the sea urchins that we've stained with a surface marker. And you can even see the tails, the cell bodies. So not only is this good for my cells, but it's good for a lot of different cell types, a lot of different potential models. So we've had a lot of fun using it, and a lot of our pictures were taken by students, which is a really great experience for them. An opportunity to work with this pretty fantastic microscope.